my time doesn't enough for doing the chromatography and extraction, isolating only the compounds by Malditov and everything. So that's why I'm not conducting this part of the characterization and standardization. So, but in the future, like inshallah, if I continue with my PhD, so this part with the thing that you mentioned, so it's gonna go under the characterization with the standardization by the chromatography.
Okay, said well. Okay, you have a good explanation. Sana, one thing I want to ask, you notice or not, how many essays have you done total in all these experiments? How you plan that?
Sorry, doctor, like just...
How many essay, this kind of beta
okay.
glucan essay, how many essay actually? Do you count it actually?
Yes, doctor, like quite a lot of the essay like going on, it's a calorimetric assay. So first of all, like I will spend 3 months of my, inshallah, 3 months, 2 to 3 months after, until like done with my characterization, extraction characterization. So that's why like I need a lot of time to do all the calorimetric assay. Yeah, I have a lot.
Yeah, you got this also. I think this one, because I'm not saying to look down on you, because as you mentioned, you keep mentioning you are mixed mode student, so you don't have really a lot of time to do this kind of thing. So you might don't want to too ambitious. Yes, you can finish, it's good thing, but too many essay also inside there. You notice also, right? Is some things cannot finish and look not good on your. This is proposal only, okay? I think you will see during the progress one. After that, then you can identify what you can achieve, what you cannot achieve, all this kind of thing. But now I think too much, but seem okay also lah, because I would think some of the essay is simply direct also essay, DPPH, something the essay is not difficult essay also. Only on the macrophage and cell culture one. That one, have you experienced before using the cell culture before?
Sorry, doctor.
Have you experienced dealing with cell culture before?
Honestly, no. Only like in the first semester, we have a lab with Dr. Ong. So I have like basic and fundamental information and how the cell cultures gonna go. But I'm so like I'm so lucky. So I have my colleagues, the lab mate. So they are working on it. So I will observe their working and how they are dealing with the cells while like conducting my other parts of the research. So I have to like get some basic skills and how to deal with just like finishing my immunoassay part, cell-based assay part.
Okay, I think overall it's quite good, but I think I agree with Dr. Venu. You need to tie up your objective, your methodology, everything aligned together. So when you do a conclusion discussion, can align together. Just make clear which one to which one. I think yours is quite clear. I think this slide also methodology quite clear also. Characterization which slide to do, but just to tidy up the things. Overall, is I think good proposal. I think good luck everything.
Thank you so much.
That's all.
Thank you so much.
Thank you so much, Sana, and thank you so much to examiners, Dr. Lai and Dr. Venu. So Sana, you can discuss with the supervisor, and I'm sure you're gonna do better. And good luck for your project. So I pass the session to Dr. Ong.
Yes, thank you, Dr. Shaida. Okay, let's continue. Next presenter will be Jing Tao Pan, student of Dr. Chung. The evaluators here, the examiners. May I know who are the examiners?
Dr. Eugene, and I can't remember the other one.
Dr. Eugene and Dr. Tasrik, he is already on the line. Kalau tak silap, wait. There. Dr. Tasrik and Dr. Eugene. Tasrik is from PJJ.
Yes.
Okay. Okay. So if everybody is here, then let's start. Go ahead, please.
Okay. Everyone. What?
Sorry, is Eugene here? I think Eugene is not here. You can better call.
Oh yeah, Eugene hilang pergi mana?
Oh, Eugene is not here. Okay.
Yeah, Eugene is not here.
Who is going to call him? Am I going to call him or?
Let me message him.
Okay, Dr. Chong. Okay, thank you.
Okay, so morning everyone. My name is Jin Tao Pan and my supervisor.
Wait. Sorry, Jin Tao. Dr. Eugene is not here yet. So we have to wait. Mm.
Oh, okay.
All right. All right. Dr. Eugene is here now. Dr. Eugene, okay, you are ready, yeah? All right. Jin Tao, you may start now.
Okay. Hi, morning everyone. My name is Jin Tao Pan. My supervisor is Dr. Chong. And my presentation topic is pentapeptide identification for the inhibition between cogranule BS189 proteins by in silico approaches. There are five parts of my presentation. First one, from 2016 to 2023, there are 370 new drugs were approved by FDA according their chemical structure. The number of peptides and oligos occupied 12.7% of new drugs. Despite the rise of small molecular drugs in 7 years, peptides still occupy an important position. So, short peptides are close effect products with high purity and more bio-friendly as bicycle biology molecules. However, they have some limitation, such as short half-time and low oral bioavailability. And we have some response methods, such as terminal protection and using natural amino acids can solve this problem. For those reasons, we consider using peptides to target surface disease. The short half-life would provide the benefits of safety. For example, a popular anti-wrinkle string, matrix 3000 contains 1 4-members peptide and 1 tripeptide. And peptide can also used in ocular disease, targeting the CNV, and one pentapeptide used to simulate one water channel protein for tears to release dry eyes. So the peptide drugs is important, but how can we design or screen the treatment peptides? There are 4 ways to build peptide libraries. First one is chemical methods. When putting new amino acids into reactor, the FMOC protection groups keep n side of amino acid cannot form peptide bonds with other amino acids. So the amino acids only form peptide bonds with n side peptide, which link with solid surface. So the T-bags and solid phase peptide synthesis both given a surface to peptides attaching and prolonging. And OBOC is for produce more huge and rounding library, because after they prolong the short terms and they miss and split them so you can get the huge and rendering library. The second one is the biology methods. Biology methods is to produce the DNA library first, and then put them into phage for merging with phage's DNA coding code protein. So we can easily use the so those coding protein contain our potential peptides. So we can easily use targeting receptor to select our targeting peptides. And then the last one method is the virtual screening. It can is a reusable and convenient method. It can contain any library size, but still require experiment verification. So why targeting S100A9 amylose is meaningful? The S100A9 monomer looks like M shape when in homodimeric situation, including 4 helix and 3 hinge regions. And S100A9 easy assembly to amylose, not only self-assembling but also assembling with other members of S100 family. And S100A9 amylose cause lots of disease, such as retinal toxin, Alzheimer's disease, and cancers. So S100A9, which is abundant in ginseng and can spontaneously form amino structure, is involved in retinal function and dysfunctions. And S100A9 amylose is the attachment core of A-beta fibers for progressing of AD disease. They have different kinds of structures, can be hydrophobic tempering and coating or low-state interaction with A-beta fibers. And this picture shows the S100A9 on the surface of on the A-beta fibers. So how to screen the treatment peptide targeting S100A9 amylose? There are 3 S100A9 inhibitors can block the can block the amylose aggregation of S100A9. The ApoE is 299 amino acids longer than the S100A9 homodimer, which is 228 amino acids. Therefore, it appears to be a bigger clip used to cap the S100A9 homodimer in solution and restrict its extension. And research of this one, the polyomentalities design the sections of S100A9 from LYS 50 to LYS 40 as have a high amino propensity. And they describe the S100A9 amino connect like occurs between both ends of each S100A9 homodimer. You can see this picture. And it was mentioned that only OLA, a natural oxidation chemical in olive oil, has the ability to disaggregate S109 fibers on the basis of blocking the elongation of exciting S100A9 fibers. And OLA shows peptide stick with TRP-80A, hydrogen bonds with ARG-85 and GLU-52 of S100A9. The interaction with S189, which include the it includes the high amino prospective section from LOS 50 to LOS 54. So the ability to discrete may be the result of an interaction force with the area from ARG 85 to TRP 88. So we think this area from ARG 85 to ALA 89 and from LEU 49 to VALO 58 of S189 is active area for S189 aminos. So this research screened 100 billion peptide phage display library against CAR proteins. It plans that the peptide can form highly interaction with S189. It can detect a concentration of core protein around tenfold lower than the mean found in a string of healthy persons. So choose the peptide to be a new drug is available. And my purpose, hopefully one day after validating the pentapeptide tree ability, the pentapeptide drugs can load on contain lessons for long-term treatment to tackling the like the acute prevention with conjunctiva. And furthermore, therapeutic pentapeptide may be delivered into brain from eyes to treat the AD disease, such as they deliver the nerve growth factors. My research aimed first to construct a 5-member peptide library for virtual screen. Second, to identify the pentapeptides like binding with S189. The last one is to elucidate the interaction between pentapeptides and S189. So it's my methodology. Some 3D structure of molecules will be downloaded from the protein data bank and chemist by the database. And ODA will be my positive control to compare the binding ability of peptides. And produce the pentapeptide library by RDKit Python API. It can transfer the peptide sequence to PDB form files. And the open-able will be used in converting the PDB to PDBQT fonts, because the PDBQT is the special font used in Autodoc. So binding free energy and hydrogen bonds will be analyzed and then ranking the Autodoc results. So we will choose some potential pentapeptide, and those more detailed analysis will be used the AMBER 2080 to do molecular dynamic simulation to discover. We can take the RMS, DRMS, and other interaction analysis. This one is my flow chart of this study. So we build the pentapeptide library first, and at the same time found out the active sites and found out the bicycle pyramids. We use the docking, and then we use the peptide selection by docking tools, and then choose the potential pentapeptide, do the MD simulation, and then do some data analysis. The last one is the sys write up and submission. So at the same time, the exploration calls for batch process to handle the huge data. The next is my Gantt chart. I will spend 2 months for peptide library inspection and 2 months for peptide selection by docking, and 2 months for MD simulation. At the same time, do some data analysis and sys write up. The last slide is my reference. I think that's all. Thank you.
Okay, thank you, Jintao. Any questions from evaluators? Of course, maybe we start with Dr. Tashrik. Dr. Tashrik.
Hello, hi. Thank you, Jinto, for your presentation. All right, okay, I have few questions regarding your project. Can you hear me?
Yeah, I can hear.
Okay, all right. Okay, for because this project you use the pentapeptide, means five member of the peptides, okay, or protein. So why did you choose this pentapeptides for this study? What the advantages do they offer over other type of peptide or molecules? Why you using pentapeptide? Why not octapeptide? Why not tripeptide? Why you choose pentapeptide for your this current research?
Because those tripeptide or two-member peptide, they are too short. And from this, sorry, from this article, they have the 18 members peptide. So they already show the peptide has a good ability to binding with our receptor. So but if we choose the triple peptide, I think it will not to give us two more binding ability to our receptor, and tripeptide library will have the 80,000 peptides. The four member peptides will be mass 20, so it will be 1,600,000 peptides. And about our pentapeptide, including the 3,200,000 peptides. So if we want to run because we think the pentapeptide library will be suitable for our project. It's not be too short and will be not be too long.
Okay, I understand what you're answering. Okay, so actually for the practicality lah, in this research you're using pentapeptide because you want to screen more.
Yeah.
More, and then you can get vast or big library, and then you can use again this as 100 A8 or A9 lah.
Yeah.
For test. Okay, all right. But because when I look on your methodology, okay, for this constructing of pentapeptide library establishment, okay, I cannot you not mention in this research methodology how many library that you're expecting to generate, to produce and everything. So I think and then also you just mentioned about you using the Python library RDKit. However, you're not specific what actually the steps that involve, like maybe you need to submit the sequence, okay, and then the sequence the RDKit will create some sort of the 5 member of peptide, and then maybe it will based on whatever lah. So I think they are lacking of the information for 3.1 in your thesis lah, and in your proposal. Okay, so I think you need to look back on this section, okay, and then try to improve lah by providing more information about how you can use this RD kit to develop or establish the virtual library.
Yeah, I told before, because this RDKT is one Python API, so I can use this one, convert the sequence to pentapeptide library. To build the three-dimensional structure of these peptide molecules.
Okay. Yeah. I think you know lah more. But yeah. But please mention it more clear lah in your proposal or on your thesis after this lah.
Yeah. Thank you.
I think Maybe we can give back to you, Dr. Eugene first, and then Dr. Eugene, and then
Yeah, no problem. Yeah. Okay.
okay.
Dr. Eugene, would you like to
Yeah.
start the? Yeah,
Okay.
thank you.
Yeah. Thank you, Dr. Ong, Dr. Ashrik. Good morning, Jintao.
Morning.
So, yeah, looking at it from, because you mentioned that it has already been screened, right? There are some in the slide 18. You showed that other researchers have also shown, have identified some peptides. I think the next one, right? Slide 18. Yeah, so is this the exact same target that you are going to screen against?
Oh, no, no, no. It's just an example. I just want to show the peptide is available very can very strong binding with my receptor. Because this one is for to detect this peptide. So you can see the longer peptide is binding in the outside of this protein. So it's not my my active site for void amyloid. You can see this from is from I don't show like because it's in the outside, but you can see this picture is in is between the two helix in the middle of like
Okay.
yeah. So they are different.
So that means that you will be docking on a specific targeted area and not the whole protein.
Yeah.
Okay. And this active site has been shown to be the interacting site between the two monomers.
Yeah, because those inhibitors was published before they just show the potential active sites only in this region, like I said before, from 85 to 88 and from 50 to 58. Yeah.
Oh, okay, thank you. So that means that if you are able to block this region, then the activity will be lost for these two proteins.
Yeah. But actually, in my view, I think this one the polyaromatic they say the active sites only only two sides of the S189, but I think it's maybe wrong. They just broke the connection. So this reason why they cannot disaggregate the performance S189 fibers, but the OLA one can. So I think I want to find the peptide can binding in these two potential active sites because ODA including the 85 to 88 and the other active sites the 52, but this one poly ultimately is only binding the 50 to 54.
Okay.
Yeah.
But in your study, you will not be using the S100A8, right? So you are only focused on A9?
Yeah. Because this example, they want to targeting the components between S100A9 and AA. But this inhibitor only targeting the S100A9 amylose, only S100A9, no AA.
Okay. So targeting A9 alone may lead to maybe some kind of therapeutic use later on, right?
What mean therapeutics?
So I mean, targeting A9 may, in the future, potentially reduce, for example, retinal toxicity. Is that correct?
Yeah.
Okay, yeah. So the other question I wanted to ask was about the pentapeptide, which you have answered as well. So back to Dr. Tashrik.
Okay, thank you, Dr. Eugene. Okay, I have one question. Can you discuss how you use Python in your research? Because from your flow, okay, this. Okay, exploring code for batch processing. But in your proposal, you not mention about how you use the Python and you also not suggested any codes that maybe you can use for this process. So can you explain how you use the code for batch processing using the Python? Generally lah.
How to say that? um, at, actually, it's a simple things. You just need to write some circles. The big circles including some small circles, and then you can do like autocomplete.
Okay. So this Python you're using what platform? You're using terminal in Linux or Ubuntu or what? Or
Anything can is based on this API can support some platform because there are some API only can use in Linux and some only can use Windows. But at this project, I think if I don't use the docking API only can use Linux. But other one I test before they can use in the Windows. It's okay.
okay. So actually I think better if you put in your proposal you can specifics the task that this automated Python script has been done lah. Because you not mention because you need to process all the batch all the data after you got from the library virtual library, and then of course because like example after you got few billion I don't know lah few structure of pentapeptide, at least you need to have the Python code that maybe it can filtering what the criteria that you will choose for the next for the docking. You will not dock everything from the virtual library, I think. So at least in your proposal, you at least you need to show the step lah, the Python code that you will be use and everything lah. That's make it clear lah. Because now you just put in the flow chart. You mentioned that exploring code for the batch processing, but in your proposal, okay, I didn't found any section that this the code, this the step and everything. So I think that maybe you can include more lah in your
I know you mean, but I just think if I show my code, it's difficult to.
Oh, okay. No. I just mentioned that maybe you can put like example, uh, data processing.py. Okay, this code, and then you mentioned that this code will using for the process all the peptide that generate from the virtual, and then give based on this criteria for the further processing with Autodoc, Vino or Autodoc 4. Then we know lah that you have this type of Python script and everything, the step and everything. So that's my suggestion lah, in order for us to look more clear about your project lah.
Oh
Because,
yeah.
I
I think in the next progress presentation, I can show you my strategy of the build the library.
Okay, all right. Good.
Okay,
And
thank
then,
you.
I think wait okay. Let's say if you finish everything and then you got the peptide that you potential peptide and everything is bind at the active site or maybe passive site, I don't know lah. So and then this can prevent from the aggregate of this S 100A9. So actually, how will your research contribute to the field of peptide drug development? So in the future, what actually this data can provide to the scientific community? Any idea?
Sorry.
Okay, let's say you finish, you identify the RMSD, you identify the interaction residue and everything, and then it shows that this amber simulation, it can block the aggregation of this S100A9. So what actually is contribute to the drug development? What the future direction that?
Oh yeah. It I think that's why I choose this project, because I want to target the surface disease. So we just deliver to your eyes and we can put in the drop and drop in your eyes. It's okay. So it's not too difficult for me to deliver the drug to my target disease. But actually, this S189 also have in some cancers lay in the middle of your body, in the deep side of your body. But in this case, I just want to deliver to to solve some eyes disease first, and then maybe someone can have some deliver strategy and deliver my peptide into the deep of the body.
Okay, alright. So you need at least after this maybe the future direction there are researcher that can use this peptide conjugate with anything and then deliver it into maybe cancer cells and then see the effects directly lah. So I think that's the future direction lah. Okay. So also you need to mention lah for the verification or validation by the in vitro or experiment lab for future direction lah. Alright. Okay, I think that's all from me lah, Dr. Eugene and Dr. Ong.
Yeah, I have no further questions also, Dr. Ong. Thanks.
Okay, go ahead, please. Dr. Eugene, do you have any questions?
Yeah, I don't have any other questions.
Oh, you don't have any? Okay, then we stop here for Jing Tao Pan. So thank you very much.
Okay, thank you.
For that. And thank you, Dr. Tashri and Dr. Eugene, for your comments and suggestions. All right, now I pass to Dr. Chong.
Okay, thank you, Dr. Ong. So we shall proceed to the last presenter for this morning session, Song Jia Yi. Because I was not given the evaluator's name, may I know who's the evaluator for Song Jia Yi?
Dr. Eric, and Dr.
Yeah.
Khoo Bunyi. Yeah.
Dr. Eric and Dr. Khoo. Okay, so Song Jia Yi, you may start your presentation now.
Okay. Good morning. I'm Song Jia and my supervisor is Dr. Ong. My research project is elucidation on mechanism of antiproliferative properties of Camelina communis against cancer of rich cell lines. Camelina communis, also known as Asia dayflower, is an annual plant native to East and Southeast Asia. And there are a variety of active ingredients such as flavonoids and alkaloids. These ingredients have many pharmacological activities, such as anti-inflammatory and antibacterial, antioxidant, and antiviral. In traditional Chinese medicine, Camelina communis is widely used for clearing heat, detoxifying, and reducing swelling. Studies have shown that different species of Camelina have shown significant activities in pharmacological. This part will focus on the research of these plants in cancer. For example, the methanol extract of C. benihalasis has antiproliferative effect on leukemia-targeted cells and achieves G1S phase arrest by affecting K-cycling. Additionally, it also has anti-cancer effect on oleic acids mouse model and shows cytotoxicity and apoptosis induction in human cancer cells, including MDMB-231. Methanol extracts from other Camelina plants also showed antiproliferative effects on oral cancer cells and are able to induce apoptosis and DNA damage. Although Camelina species plants have shown extensive potential in anti-cancer research, the specific research on Camelina communis is still limited. So the originality of this study lies in the fact that extracts from other Camelina species have shown antiproliferative effects on various cancer cell lines, and these plants have been widely used in traditional medicine, with proven pharmacological activities. The presence of these chemical compounds provide a theoretical basis for the potential of C. communis in cancer treatment. So this study is to explore the mechanism of communals inhibiting cancer cell growth and fill the gap of existing research. Best of both, the research objective is to evaluate the anti-proliferative effect of C. communals and explore its mechanism of regulating proliferation in cancer-already cell lines and non-cancer-already cell lines. The research question is, does the extract of C. communals have anti-proliferative effects on cancer-already cell lines? And by which mechanism does the extract of C. communals inhibit the proliferation of cancer-already cell lines? Is the aqueous extract of C. communals cytotoxicity to non-cancer already cell lines? Are there differences in anti-proliferative activity of C. communals among different types of cancer already cell lines? And the experiment flowchart as follows. I will start from plant collection and identification, sample preparation, extraction, standardization, and the cytotoxicity assay. If the cytotoxicity assay meets my expectation outcome, then cell cycle detection and apoptosis detection will be carried out, and then we'll do the data analysis. In methodology, because water decoction is widely used in traditional Chinese medicine, and it is a relative mine extraction method. And consider, so I will, after plant growing, I will boil in the extraction for 30 minutes. And considering the impact of solvents on cytotoxicity, this experiment will use water as a solvent for boiling extraction. After extraction, standardization tests will be conducted to ensure the consistency. The cell lines will include the breast cancer cell lines, MDMB231, and the MCF10, and non-cancer cells, MCF10A. About the drug concentration will be diluted twice from 200 to 0.75 micrograms per milliliter, and the cell cytotoxicity detected after 24, 48, and 72 hours. If the MTD results meet the expectation, and the cell cycle and apoptosis detection will be carried out for the mechanism research. Because considering that other plants of Camelina have been proved to have the effects of cell cycle arrest and apoptosis, so I choose this test. About the cell cycle test, we use PI staining and flow cytometry to measure the disruption of cells at different cell cycle stages. And the caspase cause caspase protein family plays a key role in the process of apoptosis. And its increased activity is one of the hallmarks of apoptosis. So I will choose fluorescence assay to do the caspase assay. And best of all, my expected outcome is the aqueous extract of C. communals can inhibit the proliferation of cancer-already cell lines. And the anti-proliferative effect of aqueous extract of C. communals may be achieved through inducing apoptosis or cell cycle arrest in cancer-already cell lines. The extract of C. communals is non-toxic to non-cancer-already cell lines. Milestone is I plan to in May obtain a crude extract and a standardized spectrum. In July, obtain cytotoxicity test results. August got cell cycle test results, and September got cell apoptosis results. October do the data analysis. And this is my Gantt chart. We use 1 month to do the plant collection, identification, extraction, and HPLC. After that, cytotoxicity assay and the cell cycle apoptosis each part will use 2 months to do it. If all goes well, I will finish my experiment in September, and October will do the data analysis. That's all my presentation. Thanks. Any question?
Questions from evaluator. Maybe Dr. Khoo, do you have any question to ask Sung Jia-yi?
Thank you, Prof. Chong. Jia-yi, can I see your milestone again?
Okay.
Only 6 months.
Yes.
Are you sure?
If all goes well, 6 months should can.
This one only. Yeah. You sure you have only 6 months, to complete everything?
Yes, cause I already got the cytotoxicity preliminary results and it's good.
Yeah, this is not a question, it's just to confirm with you the timeline, because 6 months, you may not able to finish everything.
Okay.
Can I comment on your proposal? Do you have the proposal with you?
Proposal review. This part literature review.
No. Do you have proposal with you now? Yeah, I know you put everything on the slide, but can you see the abstract? Just in brief. So your study aim to evaluate the effects of this plant on the cell cycle proliferations. But in terms of the write-up, I think you are not consistent, because when we go to the when you go to the objective, you see the objective in the slide, same thing. You use another statement to evaluate the proliferative effect and explore its mechanisms of regulating the proliferative effect in cancer origin cell line. I think this statement sound better. Okay.
Okay.
Yeah, so I think make it consistent, because you are not in the mechanistic study yet. You study you try try to study the mechanism related to the proliferative effect, which are cell cycle and apoptosis only, right? As stated in your proposal and also the presentation just now. Okay. Right.
Okay.
So focus on this few things, proliferative effect. I'm not sure your entity assay going to determine proliferative effect or just to assess the viability, cell viability. Maybe can you explain?
Explain.
Your entity assay, you state you want to study the proliferative effect, but you use MTT to assess it. So your MTT assay, you did mention this is viability test. So whether this test determine the cell viability or cell proliferations, can you explain to us?
About MTT, I also said the control group and the blank group, and the water extraction dilution will start from 200 micrograms and value twice to 0.75, and each cells will plant 3 batch. Each cells will do 3 plays, and for 24, 48, and 70 hours, and then can get the results.
You are explaining the entity assay, right?
Yes.
So this assay, listen properly again. This assay determines cell viability, or it will provide the cell proliferative effect to you.
It provides the cell availability to me.
It's okay. Maybe you can discuss with your supervisor later. Can I see your objectives in your slide? I think you have this specific objective in your slide. No, these are research questions.
Or this part objective.
This is objective. How about specific objectives?
Specific.
Maybe in your proposal, right? Okay.
Yes.
So in your proposal, you list down 4 specific objectives. First is to prepare aqueous extract. This is not objective. This is methodology. Okay, so please remove it. And then the second one is to assess anti-proliferative effect find. And then the third one is to identify cell death pathway. Cell death pathway. Yeah, so cell death pathway. What cell death pathway do you mean? Cell cycle, apoptosis?
Yeah. I will use cell cycle and apoptosis to this.
So please be more specific. Just don't use some other words. Just focus on cell cycle and apoptosis.
Okay.
Okay. So remember cell cycle and apoptosis.
Okay.
And then one more thing in your proposal. Refer to your proposal again. Expected outcome in your proposal. This time you did mention the apoptosis, but you miss out the cell cycle. See your proposal. Expected outcomes.
Expected outcomes.
Page 14. Page 14. I will send the review proposal to you. The proposal I have reviewed, and I put my comments back to you. Okay, later.
Okay, thank you, doctor.
All right. One last question. In your proposal, page 15, you have planned for future work. What plan of future work you want to carry out? Because your current work has not started yet, right?
Yes, have start.
Yes. So
500
is that necessary to include plan for future work? Page 15. 15. Yes. Plan for current work, I think. Plan for current work. That means your proposal. Because you don't even finish your current work, so how would you plan for your future work? Yeah. Okay. So maybe you can try to review your proposal and send that to the office later. Okay. That's all from me. Thank you very much.
Okay, thank you, Dr. Cool. Question from Dr. Eric.
Thank you, Chairman, Chairperson, Sonjay. Okay.
Okay.
Overall, it's good. The proposal. Just want to know what the research the expected research outcome. I think is this common plant have been used for other research. What's your research gap or outcome? Want to fill the gap of research in other research? What you want to achieve for your research? Can you explain more?
Cause previous research is focused on the other plants of Camelina communis, but cancer research on this communis is not is limited, and they are just focused on the anti-inflammatory, antioxidant, do not do the anti-cancer research.
One thing I think this slide also in expected outcome, I think some spelling mistake also. One thing is a cancer original cell line. What does it mean? What does it mean? Why did you mention the specific name? Why is mean like cancer original cell line? Can you explain more?
Is this plant can have work to the human cancer cells, such as cause this proposal, I choose three cells. One is non-cancer cells, and another two is to MDMB 231 and the MSF7. So I think this all are two cancer-rich cells.
I think it's better, yeah, that you spell out the cell line or you name the cell line rather than, because this is unclear because you put a cancer origin cell line or non-cancer origin, people cannot. You mean the examiner or who want to ask the question, they don't know how to ask also. Because it's a very general statement. So it's better that you put a specific name. At least they can check, okay, whether this cell line have been checked before, maybe what FCS7 or other thing, what non-cancer cell line. It's better to name it that compared to this one non-cancer cell line. We don't know what's non-cancer cell line. At least you name it. Or you want to put a specific name there, after that you put it's non-cancer cell line, it's okay. At least we can check. Now we cannot check what's non-cancer cell line. It's too many also, right? Cancer original cell line, I saw too many cell line. I don't know what you want to compare also. Okay? I just now listened that you complete the cytotoxicity already, right? The first milestone? Can you go back to you said to Dr. Koo that you complete the gun start, go to gun chart there. Yeah. You said you complete the cytotoxicity already.
Just go to preliminary results.
Oh, the preliminary results. So you complete the SPLC, you complete the plant extraction already?
Complete this part, this haven't complete.
Okay. So SPLC not complete yet, but cytotoxicity, you carry this part of the cytotoxicity study already lah.
Yeah.
Okay. Okay. So so far the progress is on time. But okay, I think overall I don't have questions. But I think just to same like Dr. Koo also, I think you make sure you have the research is in line with your objective also and expected outcome also. I say the objective also, you don't only put a general objective on the presentation. I think you should list out all the specific objective also.
Okay.
And as you know, the specific objective might be smart. I think the smart word, I think you must be measurable, all these kind of things. I think quickly, I think research objective questions or this kind of thing, you should put objective and followed by specific objective. So people can follow the study. Okay?
Okay.
I think the cancer origin cell line, you should put it somewhere lah. Some replace it with a more specific name that we can compare. Let's say breast cancer cell line, you know, at least you put a breast cancer cell line, then we know what is it. You don't know cancer, I don't know which part you want to study as well. Okay? Be
Okay.
more specific because you do final project, so it's not a very general thing. Unless you're not, you cannot determine at this point lah which cancer cell line you want to do. Then it's okay lah. If you determine which, let's say breast cancer cell line, colon cancer cell line, this kind of thing, then you stated up and the things at least we can do a comparison. And this one, at this point lah, maybe you still not carry out the research, so still okay. But I think during the progress time, I think you should spell out. I think things. Okay.
Okay, thank you, doctor.
Okay. How you want to you go back to the methodology again? Next two slide. Next slide. Okay, methodology. Next. Okay. This one, number 7, slide number 7. Okay, this boiling, you use grinding, boiling, standard of 10 standardizations. Okay, at least your standardizations. C capsules. Okay, but here you got MTTSA, you got MDA, MCF, NCF, all this kind is a cancer cell line. This is what? What is it?
What? Sorry.
MDA, MCF7, MCF10A. This is cell line you use, right?
Yes.
So
MDMB 231 is TNBC cells and MCF7 is ER cells. And M710A is breast personal cells.
So you name the cell already. So I think better when you put on objective, it's better you change to the name lah, breast cancer cell line, these kind of things. Or compare. Okay, that's all for me. Chairperson, thank you.
Okay, thank you, Dr. Eric, for your comments, and thank you again, Dr. Koo, for the comments. So we shall now end the morning session for the presentation. The evening one, we will start at 2 o'clock, and the session will be chaired by Dr. Sasi. So with that, thank you for your attention. See you in the afternoon.